Good morning. Welcome to Los Angeles. My name is Alex. Welcome to a day in my life as a creatine deficiency researcher. I'm currently a third-year PhD candidate at the Lipshutz Lab at UCLA. We study CCDS through a series of humanized cell models, starting off with stem cells, and then neurons, and organoids. And what this pathway allows us to do, it allows us to look at, one, the stem cells of the patients, which are the dividing cells. So we can look at how these cells change from being undifferentiated to turning into neurons, where we can look at 2D neuronal growth assays and characterize the neurons as well. And then using the neurons and the stem cells, we can turn them into organoids, which represent mini brains, which are a clump of cells using all three types of the neuronal brain tissue, where we can actually map the outside of the cortex within the organoid to match early human embryonic development. With these lines, what we're first looking at right now is characterizing the lines. Not much is known about CCDS in humanized models, and by comprehensively characterizing these lines, we'll be able to better create a human phenotype. What we're also looking at is testing various therapeutics with these lines. Currently, the treatment regimen has been creatine supplementation, L-ornithine supplementation, and sodium benzoate supplementation. What we're going to do is we are going to test various levels of all three of these therapeutics on our lines, and we're going to see how that relates from the patient lines compared to our corrected control lines. What we're also going to do is we're going to test a new series of adenovirus associated vectors, or AAVs, seeing how a gene correction changes our patient lines compared to our mutant lines, trying to find a curative therapy. We're starting off the day with immunohistochemistry. And what we're doing specifically is we have 4 different slides here of all organoids from day 150. And what we're doing is we're going to probe them with antibodies staining for specific proteins. And we're looking if there's a difference in neurodevelopment within the organoids towards a later stage with the antibodies. This is Rachel. Say hi, Rachel.
Hi.
We're going to the gene editing core to do our cell lines, and we're also going to look at how our images came across over the weekend. I'm all gowned up, ready to start. Well, that was more of an easier cell culture day. We changed the media on our line, we passaged it real quick, and we did a quick counting assay on the cells in the suspension. We also ran an automated plate reading for the cell confluency, and we're gonna go check that out now. Unfortunately, because the software is proprietary, I can't show you the confluency, but I can show you some representative images. What you're looking at now are stem cells that we're currently culturing in the gene editing core. And you can tell they're stem cells by the shape of the colonies, and we could see that there are 0 spikes forming on the edges of the colonies, implying that this is a very healthy stem cell culture. What we do next is we plate these cells in a 96-well plate. We then put it in a microscope that does an automatic image analysis over the course of 48 hours to a week. Shown in pink here, we then put it into a different software where we train AI models to trace the edges of the colonies. What this shows us is total cell proliferation, which means the total cell growth. We can then map that compared to the mutant lines and different other patient lines to look at representative cell growth. Today's a special lunch day. We have 3 graduating seniors and 1 transitioning grad student. Good evening. I just got home from lab. It's been a very busy afternoon. Unfortunately, I couldn't document some of the things that I did, so I'll just describe them to you here. The first thing that we did is we continued with our cell culture from the morning, where we took one of the wells that we had, and we added a solution to lift it off the plate. Using that, we plated specifically 500, 1,000, and 2,000 stem cells inside of every single well of a 96 well plate, so around 300 total wells, using a specific machine called a micro drop. So it dispenses a specific amount of fluid in each well. Using that, we set those plates in an incubator for an automated image analysis. So there's this really cool robot that goes into the incubator, it grabs the plate, brings it to the microscope, and then every single 8 hours, it will image the plate for the next week for us. What we're hoping to look for through this specific assay is we're gonna get the proliferation of the cells, meaning we're gonna look at how the cells overturn and change over time. This is incredibly important for these series of disorders because what we found so far in the organoids is that the organoids are more stem-like at a later stage compared to our corrected lines. And what we're hypothesizing is that there's altered metabolism within these cells that could drive to a faster proliferation. And this is setting up the parameters for the initial testing. We're also working on knocking out the SLC6A8 gene, and we're gonna knock out the gene and compare that to the wild type. Hopefully we can also expand to GAMPs by the end of the year. We're also using this as kind of a background for testing neurons in the future. So in the next couple months, what we're gonna be doing is adding neurons to these plates, and we're gonna be looking at how the processes grow. So the neuron has a central soma, and then there's different processes that grow from the neurons. So we're gonna look at which neurons form, look at how many processes form, and then we're also gonna look at branch points. So branch points are areas within the processes that the neurons branch and try to go to interact with multiple different neurons. So we're gonna look at the formation of those branch points over time. And we're also gonna see how the patient lines compared to the wild type lines. So we're gonna fully characterize the SLC6A8 mutations over the summer, where we're gonna look at how the metabolism is altered in the lines, how the growth rate is altered in the lines. We're also gonna look at specific organelles, the mitochondria within both the neurons and the stem cells to see how it's being impaired. And what this is gonna help us accomplish is phenotypically characterize everything about these lines. So we're gonna finally understand the unknowns about the SLC6A8 mutation within humanized stem cells and neuronal lines. We're incredibly excited to be performing this, and hopefully we'll have some updates before the conference in August. Jerry and I would like to sincerely thank the Association for Creatine Deficiency for their continued support, not only of my project, but the lab as well.
And we hope that together we can bring about a better future for your children.
Thank you.